Crimean Congo hemorrhagic fever is a viral disease. The Cchf virus is typically spread by tick bites or contact with livestock carrying the disease. It occurs in Africa, the Balkans, the Middle East, and Asia. Symptoms of Cchf may include fever, muscle pains, headache, vomiting, diarrhea, and bleeding into the skin. Onset of symptoms is less than 2 weeks following exposure. Complications may include liver failure. In those who survive, recovery generally occurs around 2 weeks after onset. The Crimean Congo hemorrhagic fever orthoneurovirus is a member of the genus Orthoneurovirus, family Neuroviridae of RNA viruses. The virions are 80 to 120 nanometers in diameter and are pleomorphic. Each virion contains 3 copies of the genome. The envelope is single-layered and is formed from a lipid bilayer 5 nanometers thick. It has no protrusions. The envelope proteins form small projections 5 to 10 nanometers long. The nucleocapsids are filamentous and circular with a length of 200 to 3000 nanometers. The genome is circular, negative-sense RNA in 3 parts: small s, medium m, and large L. The L segment is 11 to 14. 4 kilobases in length, while the M and S segments are 4 to 6, 3 and 1, 7 to 2, 1 kilobases long respectively. The L segment encodes the RNA polymerase, the M segment encodes the envelope glycoproteins GC and GN, and the S segment encodes the nucleocapsid protein. Population genetics is the genetic variation within populations and the evolutionary factors that made the variation occur. Cchf is the most genetically diverse of the arboviruses. Its nucleotide sequences frequently differ between different strains, ranging from a 20% variability for the viral S segment to 31% for the M segment. Ticks are both environmental reservoir and vector for the virus, carrying it from wild animals to domestic animals and humans. Tick species identified as infected with the virus include Hyaloma anatolicum, Hyaloma detritum, and Hyaloma marginatum. In terms of the pathogenesis of Crimean Congo hemorrhagic fever, due to a variety of factors, it is not well understood. What is known of this virus mechanism is that endothelial cells and immune cells play a large role. The occupation of viral antigens in endothelial cells is an indication that the endothelium is an important target of the virus. The diagnosis of Crimean Congo hemorrhagic fever can be done via the following: antigen capture enzyme-linked immunosorbent assay, ELISA, real-time polymerase chain reaction, virus isolation attempts, and immunohistochemical staining. Where mammalian tick infection is common, agricultural regulations require deticking farm animals before transportation or delivery. Personal tick avoidance measures are recommended, such as use of insect repellents, adequate clothing, and body inspection for adherent ticks. Management of Crimean Congo hemorrhagic fever is mostly supportive. Ribavirin is used in the treatment of this viral hemorrhagic fever via both oral and intravenous formulations, which seem effective. In 2008, more than 50 cases per year were reported from only 4 countries: Turkey, Iran, Russia and Uzbekistan. 5 to 49 cases per year were present in South Africa, Central Asia, including Pakistan and Afghanistan, and some cases in the Middle East, the United Arab Emirates. Between 2002 to 2008, the Ministry of Health of Turkey reported 128 Cchf cases, with a 5% death rate. In July 2005, authorities reported 41 cases of Cchf in central Turkey, Yozgat province, with 1 death. The virus may have evolved around 1500 to 1100 BC. It is thought that changing climate and agricultural practices around this time could be behind its evolution. During the Crimean War, the disease was known as Crimean fever and contracted by many, including Florence Nightingale. In February 1967, virologists Jack Woodall, David Simpson, Ghislaine Courtois and others published initial reports on a virus they would call the Congo virus.